Hello, Brain Shakers. Welcome to the Brain Shakers Academy. Brave Alice is here. Now, in today's session, we're going to be looking at the process of cell division. One of the characteristics of living things is their ability to reproduce and also pass on genetic information to the next generation. And they do this by going through what is known as a cell cycle. Now, what is a cell cycle? So, a cell cycle is basically a sequence of events by which a cell is going to duplicate its genome and end up having two cells that are known as two daughter cells. Now, these two daughter cells that are then formed will have the capacity of going again into another cell cycle, and then the process continues just like that. Now, the cell cycle has a number of phases. We have what is known as the interphase, and we also have what is known as the mitotic phase. Now, during the interphase, you end up seeing that the cell will go through the G1, which we call the gap 1 phase, which is this particular phase, and then they get to the second phase, which is the synthesis phase or the DNA synthesis phase, which is this S here, and then they go into the G2, which is the gap 2 phase. Then they'll go through the mitotic phase. So all this G1, S, and G2 form the interphase, and then you have the M here, which is giving you the mitotic phase. Now let's quickly look at some of the branches of the cell cycle. Now, the cell cycle can then be broken down into two main parts, which is the first one is a part or a portion of the cycle where the cell has to grow in size. It has to undergo genetic information being duplicated and being manufactured, and also the formation of relevant proteins that it is going to need as it continues with the cycle, and that is the process of cell growth. The second stage is cell reproduction, which is this particular phase here, where it ensures that it transfers all the genetic material into the daughter or resulting cell. So we have two main phases, the cell growth and the cell reproduction. Now, the cell growth is also known as the interphase period, where the cell is going to grow and make a copy of its DNA or deoxyribonucleic acid, and then goes into the second phase, the cell reproduction phase, which is also known as the mitotic phase, where the cell is going to separate its duplicated DNA into two sets and then later on divide its cytoplasm to form those two daughter cells that can either get into what is known as the resting phase or continue with the cell cycle. Now, there are phases for this particular cell to undergo. Within the cell growth phase or the interphase, it goes through a number of phases. And also when it gets to the mitotic phase, it goes through a number of phases. Now let's look at some of the phases that the cell that has entered into cell cycle will have to go through. So when we look at this particular diagram, it's going to show us the number of cycles or the number of phases that this cell is going to go through. So once a cell has been produced, it started the cell cycle, it comes into the G1 phase, which is the gap 1 phase. This is also a period that is referred to as interphase. So within the G1, the cell is growing. It goes into the S phase, there's DNA synthesis there. It goes into the G2 phase, there's a cell growth in there. So from G1 to synthesis to the G2, that is all interphase. Then the cell will then go into the mitotic phase. Now, all these subphases that the cell has to go through are well-regulated phases, well-coordinated, just to make sure that the cells that are going to be produced out of this parent cell possess the relevant, correctly and completely duplicated DNA. So the cell is going to transition from one particular phase to another phase and pass through the relevant checkpoints to make sure that everything that the cell requires it possesses before it can continue. Okay. Now, having looked at the two major branches of the cell cycle, where we have the interphase, the cell growth phase, and the mitotic phase, which is the cell reproduction phase, let's start with the cell reproduction phase. When a cell has been produced, what does this cell do? So when the cell is in the mitotic phase, it will undergo two main subdivisions or phases, which is the process of mitosis and the process of cytokinesis. Now, when it is in the mitotic phase, what is going to happen is the DNA of the cell is going to condense into visible chromosomes that will then be pulled apart by a mitotic apparatus. Now, a mitotic apparatus or mitotic spindle is a specialized structure of microtubules in which you find once the cell has begun to prepare itself, it has an organelle in there called the centrosome that contains two centrioles. Now, these two centrioles will be very cardinal in the process of cell division because they are the ones that then move to the poles of the cell and will pull in opposite direction in order for the chromosomes to be able to align themselves on the metaphase plate. Now, the process of mitosis takes place in four stages. Now, some of the literature you will read, you will find that they may classify it to be 5 stages, and basically it's because the prophase at times is classified as early prophase and late prophase or pro-metaphase. Then you have the metaphase, anaphase, and the telophase, making it 5 stages. Now we're going to be looking at the 4 main phases basically, which is the prophase, the prophase, the metaphase, anaphase, and the telophase. Now remember that we have a very dedicated session on the process of mitosis where I go into a little bit detail to explain on what happens on each and every phase of this particular type of cell division. So if you haven't subscribed to my YouTube channel, please do so. It's the Brain Shakers Academy, and look out for that particular video for you to understand the whole process of mitosis. Now, once the cell has completed the process of mitosis, we are still in the mitotic phase of the cell cycle. Once it has completed the process of mitosis, then the cell reaches a stage of cytokinesis. Now, we refer to cytokinesis as the breaking down or the separation of the cytoplasm, where the cytoplasm is going to split into two, making the two new cells, which we refer to as the daughter cells. Now, for actively and rapidly dividing cells, they may continue with the cell cycle by jumping into another cell cycle, whereas cells that do not rapidly divide may enter into the resting phase, which we call the G0. So the G0 is a permanent state for other cells, while other cells may have the capacity to restart the cycle if they do receive appropriate signals over time. Okay, now having looked at the mitotic phase, the cell has completed the process of mitosis and cytokinesis. We have 2 cells that have been produced, and we move and look at the second phase, which is the interphase. Now, the interphase is a period of cell growth, like we have already mentioned. They have the G1 phase, we have the S phase, which is the DNA replication phase or DNA synthesis phase, and you have the G2, which is the post-DNA synthesis, which is also a period of growth. Now, what happens in the G1 phase? So in the G1 phase, which is also called the first gap phase, the cell is going to grow physically larger, copy organelles, and make more proteins. It is going to build and come up with its molecular building blocks that it will need later on as it continues with the cycle, and make sure that the cell volume has been restored to its normal size. Then it goes into the S phase. Now, before transitioning into the S phase, there's a checkpoint at the G1 which controls and checks on whether the cell is of quality size, whether the cell also possesses the relevant proteins and has the building blocks that it will need, and has copied all the organelles that are relevant for the cell cycle to be completed. Then it goes into the S phase. Now, in the S phase, the cells will begin to synthesize the DNA material by making sure that it forms a copy of that DNA and also duplicates the microtubule, which is just a structure that is part of the cytoskeleton of the cell, where we were talking about the centrosome that is responsible for making sure that the chromosomes now have been correctly aligned on the metaphase plate. Then there will be transition from the S phase into the G2. Now, the G2 or the gap 2 or the second gap phase is also known as a period of cell growth, where now the proteins that were made, the organelles that were made, there is a second check that is being done on these and making sure that the DNA material that was replicated or duplicated or copied during the DNA synthesis phase is appropriate and was correctly and completely copied. Now, these checkpoints that apply at each and every stage just make sure that before a cell can transition into another phase, the correct things have been done. Now, so the G2 phase prepares the cell to enter into the mitosis phase, which we are just from looking at, and the G2 phase also ends just when the process of mitosis is beginning. Now, like we said in our beginning, that the cell cycle is ordered and well regulated. We need to look at how this cell cycle is regulated by looking at some of the checkpoints that are applicable to the cell cycle. So the cell cycle is mainly regulated by the checkpoints. Now, these checkpoints that are there make sure that the process is irreversible and unidirectional. So there are three main checkpoints to look at, which is the G1 checkpoint, the G2 checkpoint, and the mitotic spindle checkpoint. Now, with the G1 checkpoint, which is a main decision point, you find that the cells that pass through this stage may end up completing the cell cycle and forming two daughter cells. So the responsibility of the G1 cycle is to make sure that it checks on whether the DNA is of good integrity, and also makes sure that the proteins that were synthesized during the process of, or during the phase of the G1 are of quality, and if not, the cell then would be sent into the G0, which is a resting point for the cell to wait until it is repaired before it can continue with the cell cycle. If everything is okay, the cell is then allowed to proceed from the G1 into the synthesis phase, where the DNA now is going to be copied. Before the synthesis phase is then completed, you also notice that there are other important regulatory proteins which we'll be looking at, such as cyclins, playing a key role as well during that DNA replication. Then you get into the G2, where you end up with the G2 checkpoint. Now, the G2 checkpoint just happens right before you get into the mitotic phase and checks on whether the DNA that was being replicated or copied was replicated correctly, completely, and it is undamaged. Now, if there is any damage to the DNA material, the cell can then go into a resting phase. The cell is then entering into a phase where it is going to pause and await for repairs to be conducted. And once the repairs have been conducted, the cell can continue with a cell division. If the cell does not undergo repair, then programmed cell death is going to move in a process that we call apoptosis. Then we get to the third checkpoint, which is the mitotic spindle checkpoint. This one just basically checks on whether mitosis has been complete or whether the sister chromatids have been correctly attached to the spindle microtubules as the cell is going to be dividing. Now, at each of these checkpoints, the cells checks and make sure that all the tasks that need to be done have been completed before you move on to the next phase. So they are the go ahead signals. They are checkpoints to allow the cell to either proceed or to go into the waiting phase for repair. Now, this particular cell cycle is regulated by a number of proteins. Now, the commonest proteins that we do see regulating the process or the cell cycle itself are the p53, the cyclins, and the cyclin-dependent kinases. Now, the p53 is of importance in that it has the capacity also to check that if this DNA is damaged. Once this DNA is damaged, what happens? We have to protect the body by doing one, by initiating repair. So the p53 initiates repair for any damaged DNA. If the DNA cannot then be repaired, then the DNA is going to undergo programmed cell death. So it has the capacity to stop the cell from entering mitosis, ensuring that all cells that enter the cell cycle have or possess DNA material that is of great quality. Now, you also have cyclins. Cyclins are just a group of proteins that are produced at every other level of the cell cycle, where you see if you are in the G1, you find that they are G1 cyclins. If you are in the S phase, you find you have S cyclins. If you are in the G2, you have G2 cyclins. You're in the mitotic phase, you also have the mitotic cyclins as well. Then you have the third type of proteins, which is the cyclin-dependent kinases. Now, the cyclin-dependent kinases are another important group of proteins that have the capacity to activate other enzymes and other proteins by binding to cyclins, and they do this by making sure that they affix to themselves a phosphate group, and that is how they get activated to be able to then activate other enzymes and proteins that will control and regulate the cell cycle. So they usually are in an inactive form until they have affixed the phosphate to themselves. Then they are active to be able to activate further proteins that will be able to control and regulate the cell cycle. Now, after looking at the cell cycle itself, then we would be proceeding to look at a number of cell divisions. So we have a dedicated video that is looking at mitosis, or the process of mitosis, where one parent cell is going to end up into two daughter cells, and the process also of meiosis, which is basically concerned with sex cells. Now, the cell cycle is just basically the two main phases that the cell has to go through, which is the interphase and the mitotic phase. So if you like this video and you would like to see more sessions like this one, please do not forget to subscribe to my YouTube channel, The Brain Shakers Academy. Hit that notification button so you're notified every time I post something new. Like, share, give it a thumbs up, then share this video so many people can have access to it. And as always, it's a pleasure to have you around. Thank you for watching, and I'll see you in the next one.